Hi, it's Jim Lindeman again. We're on the second to last topic, which is DNA. And when I did background research for this, I ended up with about 45 slides, which I boiled down to about 17. So if you can imagine how much that there is that we could talk about, but I tried to keep it to the things that would get across to you the majesty of this molecule and the extraordinary way that God has designed it. Anyway, let's get on here. So, don't be absurd. Nobody made us. We evolved by chance from snowflakes. Well, something to think about. All right, DNA, information and meta-information, a twofold information repository. Okay, when you take a look at what the cell needs, it needs, first of all, information, or what's called information. What is each object in the cell? How is it made? How does it work? When is it to work? What does it influence? Should it be repaired, ejected, or disassembled? And how? These are all kinds of things that the cell has to know as it goes about the business of living. And so something has to be a repository of all of that kind of information. But there's a second-fold problem here as well. Okay, first of all, comprehensively regulated information-driven metabolic functions, total physical details. In other words, eye color, shape, hair color type, body type, skull shape, etc. The flora would be what kind of a plant is it? Vine, tree, moss, size, shape, leaves, reproductive system, and so forth. All of this stuff has to be available for the cell to operate. Somewhere there has to be the place where this information is stored so that it can be done or used or whatever when needed. So on one hand, you have information. On the second hand, you have to know what to do with the information, and you need information about that. In other words, what information is there in the cell? How does this information get activated? What information will be activated? In other words, as you have cells doing different tasks, say for instance in the human body, how does the cell know what it is supposed to do as opposed to what it's not supposed to do? How does the liver cell know that it's just to do the liver operations, functions, as opposed to that it's not the skin? So when does the information get activated? So for instance, as the embryo is growing, who's going to be operating now? What's it going to be doing? How is it going to be happening? Will this information get passed on? In other words, it's great to have the information, but where does it end up going? Does it get into the next generation or not? How is the information to be passed on? How is the information to be protected, preserved, so that it's guaranteed to be useful information and not just get immediately corrupted down the way? How do you have the kinds of systems set up to make this information available, good copies, things that are going to help a cell to survive, to do the job that it's supposed to do, and how is that going to get protected and passed on? So you have, in a sense, two tremendous functions that are going to be governed by this little thing called the DNA molecule, deoxyribonucleic acid. All right. We now know that the secret of life lies not in the chemical ingredients as such, but with the logical structure and organizational arrangement of the molecules. Like a supercomputer, life is an information processing system. It is the software of the living cell that is the real mystery, not the hardware. In other words, we know the hardware stuff. But how did stupid atoms spontaneously write their own software? Nobody knows. And this is the key. How did stupid atoms come up with all of this information, not only to have it, but also to protect it and to be able to pass it on? This is the problem that is facing evolution, trying to figure out how did that come into being. And remember, we talked about this in the last topic, is that you have this Polyany impossibilities, is that each layer governs the one below, but the layer cannot create the one above. And so how did stupid atoms write the software which will now govern how the cell is going to use those atoms? The DNA molecule, the information safe. Researchers have created a DNA computer made of strands of synthetic DNA and used it to solve complex problems. Much more development will be required before DNA molecules replace silicon chips. However, as 1 gram of DNA can hold the information equivalent of 1 trillion CDs, scientists are looking to use DNA's amazingly designed, and again, key word here, is that word designed information storage capacity, able to hold the blueprints of life, to overcome looming limits to microchip power. Also, as the fledgling DNA computer solve problems in far fewer steps than a conventional computer, the researchers expect biocomputers to be capable of dealing with problems of greater complexity while using less space. To realize that in that little molecule, you have extraordinary capability. The DNA double helix, just right structure for genetic information storage, beautifully matched by the elegance and efficiency of the code in which the cell's information is written. Biologically relevant information, brilliant inventive strategies and tactics for the extraordinary challenges of metabolism and reproduction in a constantly changing environment, choreographed, intricate, and harmonious regulation. The right things happen at the right time in the right sequence, else chaos and death. Biochemical molecules in constant complex movement, in intense and lightning-fast activity, up to 1 million events per second for a single reaction. To think about, this is constantly, this is always what's going on. 24 hours a day, 7 days a week, 365 or 6 days a year, constantly. Billions and millions and all kinds of things that are happening in the cells, never stopping. All governed by the ability of the DNA to control all of that and be able to push it forward to the future, the next day. The message transcends the medium. Information is not the alphabet itself, although the alphabet holds the information. In other words, you take a look at the alphabet here. The alphabet here is just a bunch of letters. Squiggles on the paper or squiggles on the screen. But it holds the information. It is not the information. The information can be held either in paper or pen or a computer drive or whatever else. The storage medium holds the message, although the storage medium is not the message. It just simply is occupied by the message. It is found in the coding and decoding of the alphabet. You know, every book you've ever read is just a different combination of 26 letters. Look at this library and the books that just go almost as far as the eye can see. And all of them, only 26 letters to the alphabet. Though only 4 types of DNA bases, A, C, G, T, arrangement is no meaningless bowl of alphabet soup. And interesting to note that the C, the cytosine, is unlikely to be found in any such soup. It is a very difficult molecule to produce. A group codon of 3 DNA letters codes for 1 protein letter, called an amino acid, and the conversion is called translation. The language is the key. For instance, you have 4 letters here, G-I-F-T. In English, it means a present. Same letters in German means poison. And so the code is very, very important in holding what the information is all about. Unless it's joined in the right sequence with an understood language, there is no usable message. Scrabble letters mean nothing until a message or word is imposed upon them. Information is the very opposite of chance. If letters are to be arranged into a sequence to spell a message, a particular order has to be imposed on the matter. And so this is a big, big problem for anything that you want to talk about in terms of evolution, about chance just happening to connect this and that and so forth. You do not have the ability of chance to come up with this kind of a result. What about the 1,000 monkeys with typewriters or computers in infinite time to type out Shakespeare? Well, in 2003, the Arts Council for England paid £2,000 for a real-life test of the theorem involving 6 Sulawesi-crested mackies. But the trial was abandoned after a month. The monkeys produced 5 pages of text, mainly composed of the letter S, but failed to type anything close to a word of English, broke the computer, and used the keyboard as a laboratory. The problem with random is that it is exactly that. In other words, if so often it seems as though the concept that's being used in evolution of random chance connecting a couple of molecules together into whatever is that it's a trial and error. But then when the error is that they don't work, the error is discarded and something new is tried. The problem with random is that that's not random. For instance, a song player may be set to random, but it really isn't random. It's an algorithm to play randomly those songs not yet played until the last remaining song is played. A true random would mean that there is nothing to stop replaying the same songs over and over, quite possibly never reaching the final song at all. So in other words, for them to keep typing S means that for the whole time they're there, maybe they're going to just type the same letters all over again. And so the theory that you're going to have by chance the ability of monkeys to have typed out the whole of, say, for instance, Shakespeare's works is not something that's a realistic projection. There is no known non-intelligent cause that has ever been observed to generate even a small portion of the literally encyclopedic information required for life. Where would it come from? As I said in the last slide, how to dumb atoms write their own program. DNA capacity. Now we looked at that library before. DNA, million times more efficient at storing information than, for example, a Blu-ray disc. Even more space is saved by its tight coils. A single human cell's DNA, visible only through a microscope, unwound, would stretch to 2 meters. In other words, if you were to take all the DNA in just one cell of your body, it would be taller than you. DNA equals 500 large books of small type words, or 130 volume encyclopedia sets, the recipe for all the cell structures, and able to, for example, detail the intricate construction of the human brain with its trillions of precise connections. Enormous information content is transmitted one generation to the next. All information required to make every unique human being could be stored in a volume of DNA no bigger than a couple of aspirin tablets. It's hard for me to conceive how this can be that potent, but apparently this is what happens. This is what's available. All living things have encyclopedic information content, a recipe for their complex machinery and structures. This is stored and transmitted to the next generation as a message on DNA letters, but the message is in the arrangement, not in the letters themselves. The message requires decoding and transmission machinery, which itself is part of the stored message. The choices of the code and even the letters are optimal. Therefore, the genetic coding system is an example of irreducible complexity. That is, the level below cannot tell how this level came to exist. The library. The DNA is coiled around protein globules called histones, then coiled again into a rope-like structure, then supercoiled into two stages around scaffold proteins, into thick chromosomes, the closed books seen under the microscope. The coiling prevents the coded information from coming into contact with the translation machinery. When information is required, a particular page is opened, photocopied, and then the book is closed again. Where to begin? DNA letters are read in groups of 3, the sequence, GTT, and all this rest. Starting from the first letter is GTT, CAA, CGC, and so forth. However, if you start from the second letter, you have a whole different sequence happening. A totally different protein arises because of the different starting. Only one letter away. Now you talk about the complexity of the DNA, the strand, is that you have genes and sequences that are multi-use. They will construct different proteins. And yet each of those proteins is essential in the running of the cell. Now, how do you have mere chance coming up with something so very highly designed as something like this? The Human Genome Project only could identify 35,000 genes about, and yet there are over 100,000 proteins that the cells use in the human body. So how do you do this? It's because you have, even in this coding, the ability to have different proteins which do different jobs come out of the same area. Therefore, DNA is an even more compact and complex information storage system. It's not simply just a book where you open up a page and you read whatever the message is that if you were to open up the same page but start in a different spot, you would have a whole different story. And if you were starting a different spot, you'd have a whole other different story, even on the same page. That's what you have happening with the DNA. The histone code, the way the DNA is wrapped, controls which proteins are made. Packing the DNA loosely or tightly, genes can be switched on or off as needed. The splicing code, spliceosomes. Elaborate machinery must be in the right place and direction on the Elaborate machinery must be in the right place and direction on the mRNA, the messenger RNA. Otherwise, the gene and subsequent protein are malformed at best, destructive at worst. In other words, if it is not doing its job exactly where it's supposed to go, how does it know that? Where does it get its information from? It isn't just mere chance. It's something that's very, very precise. Epigenetic, regulatory systems. Gene regulation by chemical groups attaching or detaching to or from the DNA, enabling different programs to run independent upon the organism's dependent upon the organism's needs at a particular time. So you have other things happening to the DNA which are activating different spots. And somehow, in the midst of all of this, is constantly controlling what's going on in the cell so that it runs at optimum level, no matter what the environment may be. 1/3, let's see, okay. Less than 2% of the DNA codes for proteins. Supposedly 98% was from past evolutionary steps and failures and was therefore called junk DNA. And evolutionists were very happy about that because, see, you have the vestigial organs, so to speak, of the cell in junk DNA. Well, just like with the human vestigial organs, what happened was, because they assigned certain things as useless, unnecessary tissue, there was no research being done upon them, which now is being done, and the more and more that's being known, the whole idea of vestigial organs in the human body has shrunk to zero. There's nothing in your body that is extraneous. Same thing here. ENCODE, the Encyclopedia of DNA Elements project, over 440 scientists at 32 institutes performing over 1,600 experiments. Although details were still in progress as of 2007, besides the 2% of genes, over 80% of the human DNA does something. It's a huge control panel with millions of switches governing the genes. In other words, you have little, for instance, you have little micro DNA, or RNA rather, controls behavior of genes. That they are actually coded and they go out and they act upon the proteins and other kinds of things. The order of on switched, the order of what is going to be switched on as, first of all, the creation of protein, but also as to what's going to be used next. Different cells have switch settings requiring different active DNA areas. ENCODE only looked at 147 types of cells out of a few thousand. Remaining 20%, in other words, of the 80%, the remaining 20% is fully expected to reveal more switches and controls. That by the time that they are done, they did not expect to have any redundant DNA or junk at all. 1/3 of the mouse genome, once thought as junk DNA, governs embryo development. The complex sequence of events changing a single-celled egg into a complex organism with hundreds of different cell types and different organs, all in the right places, and programming for this has to be somewhere. It appears to be in this supposed junk. In other words, here again you have the encyclopedic ability of DNA. And it's all stored there so that from one generation to another generation, you have the ability of the organism not just to survive, but to reproduce, and for it to reproduce, and for it to be able to handle whatever the environment tends to throw at it. The untranslated regions, transcripts, photocopies of the DNA. There's a huge overlap. One of the things that they found was that when a protein is formed, it isn't just the genes that are copied. You have a huge overlap, over 10 to 50 times the size of the gene region. 20% or even more than 100 times the size. You have a huge expanse that's being copied at a time when a gene is unfolded so that it can be then produced by or given to the RNA in order to produce the protein. So why is it doing this? It's like photographing 100 other pages to know how to use a certain page in a book. At least 93%, probably 100% of the genome is transcribed, not the 3%, not the 3% of just the gene region. You have basically all of the DNA in action when RNA is copied, when a protein is then formed. Why is that happening? The untranslated regions called introns, now called UTRs rather than junk, the UTR, untranslated region, are far more important than the translated regions, the exons, the genes. Exons, the genes are no beads on a string, but are interleaved, overlapping segments with typically 5, 7, or even more than 9 transcripts. In other words, this particular gene area will be used for many, many, many other kinds of things than just one particular protein. And the thing is that because it's interleaved, then you have not necessarily a gene as a bead on a string. You have pieces of a gene all together. Exons are not gene-specific, but modules. In other words, the things that we would call the genes, the pieces, the exons, are not gene-specific, but modules, part of a gene, which can combine with different genes located on different chromosomes. Introns are transcribed on average in 7 different ways. The introns are those parts that are not the genes. There are 33 times more bases in introns than in exons. In other words, the introns are up to 50% more active than the gene areas. Now, if this is junk DNA, it is extremely active. It is extremely involved with what is going on in the cell. And so the whole concept of, well, this is just junk, this is left over from evolution, does not hold water. Energy, etc., required means such full transcription is considered essential. You have to remember is for the gene to do any kind of this transcription from the DNA, it's work. It costs in terms of energy. Why would something as efficient as the cell, why would it go and just do things uselessly simply in order to please the idea of random choice? Then the translation goes into protein. Introns are spliced out, exons are joined up to produce the protein. All of this is happening, but all of this with precision. The spliceosomes are sitting exactly where they need to be, in the direction they need to be able to start now assembling what will then become the basis for which the protein then will be built. All of this is going on inside the cell. All of this is going on with extreme precision. Somewhere is that information. How does it get there? And how is it working? The transcripts of the DNA. DNA is like a coiled zipper. One zipper side, what's called the sense strand, the other is the anti-sense strand, has the desired sequence for the needed exon or gene. Both strands, sense and anti-sense, are fully transcribed. Both sides, even though only one is perhaps most needed. Transcription begins at a specially marked start position and ends at a special stop sign. Proceeds both backwards and forwards. Many possible start sites with each gene region, like I said, is that maybe you have a gene starts here. For the next sequence of the next protein that's going to be built, maybe it'll start here or here. And that's the thing is that, how does it know? How does it know that that's where it needs to be in order to start? It's not random, it's precise. This is the RNA polymerase, in which you take the DNA comes through, kind of split apart, and you have the RNA coming out the one side as the DNA is being transcribed. Gene switches, transcription factors, adjacent to the start end, may be thousands of base pairs away from the gene they control, even on different chromosomes. That's kind of, wow. The product is messenger RNA. Chromosomes in each cell are stored in the membrane-bound nucleus. The nuclear membrane has about 2,000 pores in it, through which molecules can be passed in or out. The required chromosome is brought near to one of these nuclear pores. The section of DNA to be transcribed is placed in front of the pore. The supercoil is unwound to expose the transcription region. The histone coils are twisted so as to expose the required copying site. The double helix of the DNA is unzipped to expose the coded information. The DNA is grasped into a loop by the enzymes that do the copying, and this loop is then copied into an RNA transcript. The transcript is checked for accuracy and is degraded and recycled if it is faulty. The RNA transcript is specially tagged for export and is exported throughout the pore and carried to wherever it's needed in the cell. The book of DNA information is closed by a reversal of the coiling process and the movement of the chromosome away from the nuclear pore region. Ironic, isn't it? Something that was supposed to have happened by chance should have as its product something that leaves nothing to chance. The precision, the absolute precision, the choreograph of all of this. How does that happen? Consider the formation of the transferred RNA and the messenger RNA. So the messenger RNA is what comes out of the transcription process. The transfer comes and gathers all the amino acids that are going to be needed, bring it to the messenger RNA, and make the protein. Where a certain segment of the DNA molecule, not too long, not too short, separates and attracts the counterpart nucleotides. The RNA doesn't even stop for T, but separates itself. Meanwhile, the DNA zips itself shut again. The smaller tRNA, transfer RNA, each hunts up only the amino acid that fits it and forms a strong enough bond to carry it to the mRNA, attaches itself to the proper place on the messenger RNA, and waits. Finally, all of the amino acids are joined to form a protein molecule. Now bonds that were strong enough to hold and carry are broken. None too soon, nor too late. Just exactly according to the timing. How very, very closely precise is what happens in the cell. Vicious circles, chicken and egg problem. So the DNA transcribed into messenger RNA by RNA polymerase, translated into a protein in the ribosomes by tRNA adapter molecules. So there's the protein. Protein is escorted by chaperones into a chaperonin so that it folds correctly. So in other words, all of this machinery has to be, first of all, encoded in the DNA, read in the DNA, made the machinery, so that then when the time comes for the proteins, it has the places it needs to have so that it can function properly in the cell. Planned supercomputer slower than real life. IBM plans to spend $100 million building a new supercomputer to simulate a basic biological process, protein folding. Proteins, for instance insulin, are assembled as long chains which must then fold into the correct three-dimensional shape in order to perform their function. As simulating this basic life process is beyond the reach of contemporary computing, the Blue Gene supercomputer is being built to run 500 times faster than the world's fastest computer today. It is expected to take 5 years before the supercomputer can be ready to tackle the grand challenge of protein folding. And even if everything works as planned, it will still take the Blue Gene about a year to run a computer simulation of the folding of a single protein. How long does it take living cells to actually fold one? Less than a second. As one IBM researcher noted, it's absolutely amazing the complexity of the problem and the simplicity with which the body does it every day. Yeah, okay, if you want to say it's by chance, it's entirely up to you. Messenger RNA, RNA polymerase, transfer RNA, ribosomes, chaperones, enzymes, all this machinery is itself encoded in the DNA. The DNA can't be read without this decoding machinery. Unfortunately, they have to be read out of the DNA first. These processes use energy supplied by ATP. ATP supplied by the motor ATP synthase. The ATP synthase motor created by instructions in the DNA, decoded only by using ATP, a three-way circle. In other words, what you have going on in the cell defies explanation as to how it started. Because the machinery that's needed to read the DNA has to exist prior and has to be basically read out of the DNA first, which means somewhere along the line the machinery had to start right at the beginning. How did the instructions first get coded and then placed into the DNA? Now, at the moment, we're talking entirely upon taking what's in the DNA and bringing it out. What could have coded the DNA to begin with? How can you have protein models or forms coded into the DNA in such a way that they overlap? There is an extraordinary requirement, at least it would seem, of intelligence in order for all of that to be planned out so that what you end up with is such a compact final product. How did stupid atoms spontaneously write their own software? Nobody knows. Where did the decoding machinery come from? Thus, the code cannot be translated except by using certain products of its translation. This constitutes a baffling circle, a really vicious circle. It seems, for any attempt to form a model or theory of the genesis of the genetic code. Optimized code. A pairs only with T, C only with G, due to the basis chemical structures. New strands will copy the information exactly, far more precise than pure chemistry. In other words, it isn't a matter of just simply putting them together and whatever you get is what you get, but rather you have built into the DNA the potential to always have an exact copy. It minimizes error. One mistake in 10 billion copies. Editing, proofreading, error checking machinery is encoded in the DNA so that it guarantees that this kind of thing happening is going to become extremely rare. So this is what's happening throughout your body every day, from the time that you were conceived from a little egg, fertilized egg. That all of this has been going on all along. Because if those mistakes did creep in, by the time you've reached your age by now, how many problems would you have in your body from all the errors that would creep in if all of this was not a reality? Irreducible complexity. Unless DNA information for an editing machinery was from the beginning without such a high degree of accuracy, error catastrophe would destroy the cell for information noise, junk protein accumulation. The Poliani impossibility. How did stupid atoms spontaneously write their own software to create a code which had to be in place to carry on to the next generation? to come up with even the concept of an error checking routine. Where did this all come from? From stupid cells, or excuse me, from stupid atoms? As a computer copies a program from hard drive to memory, so DNA must be copied to RNA. Despite the error checking, a rare error occurs, and perhaps the wrong amino acid may be subsequently placed into a position in a protein. Yet the code design is such that even if an incorrect amino acid is selected, it will often have similar characteristics, allowing the protein to do its task. Of all possible codes, this one is very good at compensating for errors, an optimized design. The standard DNA code used by the vast majority of organisms may be the very best. According to researchers, they cannot come up with something better than this. The DNA language optimized by gradual change through mutation. If keys on the computer keyboard were changed without simultaneous change in the software which reads the keys, numerous words would become nonsense. A single DNA change requires how the machinery error checks the new code, as well as how it is interpreted. Additionally, considering how specific genes can be coded for different protein strands. Virtually all organisms use the same code. This optimization must have occurred very early in the evolution story, thus severely limiting the time for such miracles to happen. Sir Francis Crick, co-discoverer of the DNA structure, argued that once in place, the DNA code would be frozen, as it would be very difficult, if not impossible, to change. In other words, one change can be so catastrophic throughout the whole cell that you end up with the cell literally ceasing to function. Even Richard Dawkins, who is very, very anti-creationist, wrote in The Greatest Show on Earth, any mutation in the genetic code would have an instantly catastrophic effect, not just in one place, but throughout the whole organism. If any word changed its meaning so that it became to specify a different amino acid, just about every protein in the body would instantaneously change. Unlike an ordinary mutation, which might say slightly lengthen a leg, or shorten a wing, or darken an eye, a change in the genetic code would change everything at once, all over the body, and this would spell disaster. Changing to a new protein, fitness landscape. Protein function in the present cell is at optimum, like standing at the top of a hill. Useful amino acid sequences are very rare, functionally isolated from one another. In other words, the amino acids that you have, the strands that you have, the proteins that you have in your body are at optimum. This is proved by the fact that your body has functioned for how many years. And for many people who it functions even into their hundreds. And so these protein strands are exactly what they have to be. If you think in terms of the proteins, the amino acid sequence, standing at the top of a hill because it's at optimum. Now if you change an amino acid sequence, it makes the protein function less well. Why? Because from what it was able to do, now it has been crippled. It starts down the hill. Somewhere the valley must be encountered before climbing up the next optimum hill. So in other words, for a amino acid sequence, it's isolated from others. In other words, it just can't simply move over. It really has to get to the point where it degrades before finally it can improve. Natural selection would tend to reject the less optimum functioning cell. It would preserve the original optimum sequence, keeping the gene at top. Why should natural selection say, oh, well, I guess I'll have to wait here before I do anything about this less optimum cell function? until finally something else comes. No, and you see, this is where, again, a problem I have so often with evolution is that it seems to have the capacity for intelligence on its own, to be able to make allowances, to plan, to whatever else. It is not treating everything with absolute random. Gene duplications is a wonderful thing that's being touted as the reason why you have additional information so that bacteria can finally become a human being. The human genome requires far more information than a bacteria genome. There is no known process to create code from nothing. In other words, how do you generate code? Okay, now you've got DNA, but how do you make it now hold something that has never been created before in terms of a function or whatever else? How do you do that? How do you reverse the process of putting information into a DNA molecule? Gene duplication provides existing code which could be modified. Evolution should provide DNA en route to a new function, although not yet useful. Therefore, there should be lots of junk DNA. This is one reason why junk DNA was so the dear of many evolutionists. Only the problem is that it's not junk DNA. There is extraordinary function, even in those parts that have no coding to create a protein on their own. How is the new function turned off until it's ready? In other words, supposedly, everything says, okay, I'm not going to bother with this, I'm going to shut this down until finally I can fine-tune it, and then when it's ready to go, then I'm going to make it now turn on. You still have this sense of, if this is dumb atoms writing their own software, why would it do that? How is the new function turned off until it's ready? Otherwise, species would experience error catastrophe. In other words, you'd have this clutter building up inside a cell until finally the cell cannot operate, because if you have all this junk DNA and junk proteins that are just sitting there filling the cell, preventing the cell from being able to continue its extraordinary, precise, choreographed activity. Genetic load. Darwinism depends on mutations or garbling of genetic inheritance, which is almost always harmful, as a means to program evolution to more complex forms. Every competent space mathematician would assure you that such a Darwinian idea had no chance of working. Every computer expert will in fact assure you that throwing random mistakes into a computer is no way to improve it. A mutation affects the order of the nucleotide pairs, say for instance in the reproductive cells. Most will be benign with no apparent effect. For instance, perhaps air color. Theorized, very few provide any kind of benefit. Some reduce effectiveness. Some will reduce effectiveness, for instance, blindness in kingfish or color blindness. Some will kill the cell outright, just absolutely prevent it from doing its job. The remaining few will be ticking time bombs having various effects. For instance, allergies, sickle cell anemia, cancer tendency, blue bloods. One of the things that I found was very interesting about sickle cell anemia is that, well, you see, this is where there's a positive result from sickle cell anemia, because there is a defense that it provides against malaria. So that therefore, see, there can be a good result from a mutation. The only thing is that as I looked at sickle cell anemia, there is advantage. The average lifespan in the US in 1973 was 14 years old. So yeah, there's a protection against malaria, except that for up to the 1973, you didn't expect to see your teenage years, or very far into your teenage years. That's an improvement. That's a benefit. And a lot of pain from tissue damage. In other words, what would happen with the sickle cell anemia is that these would all clump up in the blood flow. And therefore, it would actually cut off blood flow to certain parts. And it would lead to muscles or other tissue damage, organ damage, and that therefore there would be a lot of pain. And so you're sitting there saying, yeah, evolutionists want to talk about how this is an advantage, this is an example of a mutation being an advantage or giving an advantage. This genetic load accumulates throughout the generations, distributing weakness, some potentially lethal, throughout the population. So in other words, like with the blue bloods, what was happening was that there was so much intermarriage in the nobility of Europe that literally the blood was becoming less effective in doing its job. That's why they were called blue bloods, because it was not, the bloods were not really releasing the carbon dioxide the way it should. Therefore, you have a less effective organism. Some potentially lethal throughout the population, awaiting a trigger. Perhaps it was environmental, or perhaps both parents carry the gene. In other words, like with the sickle cell anemia, it's only when both parents carry the gene that it will manifest itself. So it stays hidden until finally that happens. Or anything else that may trigger that particular mutation, and it will do its damage. If a new species arises by evolution, what happens to this genetic load? A lot of times I think what I find is fascinating is that there seems to be, again, in the assumption in the evolutionist camp, that once you have a new species arise, it somehow starts with a clean slate. And now you have to pursue the idea of what happens when there's detrimental mutations happening within it. My contention is that if you have enough mutations to raise a new species, enough should be passed on to destroy it. It already has this load of death that's being carried around. Sure, maybe you've got an absolutely new species happen, but what happens to that genetic load that's sitting there poised, ready to destroy the previous species? According to evolution, DNA has evolved an unobserved and unprovable assertion over billions of years into this wonderfully complex, remarkably efficient, spacious depository of information. The most elegant data processing system known to man, available to even the most tiny microbe. Suddenly, for no apparent reason, it is de-evolving at an alarming rate. All genomes are slowly but relentlessly degrading through mutation and copying errors, unable to keep up with the proposed beneficial changes. In other words, even if there are beneficial, what's happening is that more and more of the genome is getting worse and worse. All genomes. 98% of species have already become extinct. No new ones are on the horizon. Why? Dog breeds come from the wolf. From the Great Dane to the Chihuahua, to the Pug, and to others. Different height, ears, fur, snout, bill, tails, feet, all already in the genome. In other words, in the wolf, is there the potential of such variation that can give rise to this extraordinary amount of variety or variation? Some of it may be from mutation, but on the other hand, not all of it can be. But one thing that's interesting is that you notice they're all still dogs. No matter what the variation is, they're still dogs. But can a Chihuahua within its breed be selectively bred into a Great Dane, or has there been a loss of genetic information? So, in other words, can you take this and make that out of this? Or do you have to interbreed? Do you have to return to its genome, to its specific genome, the ability to have that kind of variation? And so even though you want to talk about how selection and everything else like that will produce something new, the evidence that's out there is that you end up with something less. Less capable, less able, less potential variation to meet whatever the need may be. Darwin, if all pigeon breeds interbred freely, the result would be the common rock pigeon. In other words, if you restore the ability of the DNA to have the variation, then you're going to be able to start with being able to go. Does purebred indicate new species or a degraded genome, unlike the wolves? Does natural selection or selective breeding reduce versatility of the organization? Some capabilities will be bred out of the genome. You have supergerms in which, yeah, they will be able to withstand certain antibiotics, but at the same time they become less versatile. Many purebreds have genetic problems, hips, disposition. Would interbreeding now only perpetuate a defect already isolated by selective breeding? Is the net effect of the organism less responsive to the environment? Natural selection's reality. Natural selection may explain the survival of the fittest, but it cannot explain the arrival of the fittest. Alexander Graham Bell, the inventor of the telephone and student of evolutionary problems, said, "Natural selection does not and cannot produce new species and varieties. On the contrary, its sole function is to prevent evolution." What's an effective way to kill a seagull? Tie a ribbon to its leg, and it will be pecked to death by all of the rest of its flock. Idea of natural selection is to preserve, not to create something new. What would preserve the development of beauty? And then Gould, with his sexual attraction for hopeful monsters, sudden jumps like the bird from a reptile egg, again, you have those kinds of problems. The subtle anthropomorphizing of evolution, that somehow evolution has a plan, it has a direction, it has purpose, even though supposedly it's merely random. So I'm going to stop. Sorry for the glitch, but we continue here. One of the things about the sexual attraction for hopeful monsters is the idea that Gould talked about, well, okay, suddenly you have a jump of a whole different type of creature. And that supposedly, especially with sexual reproduction, its counterpart would have to also have that kind of a jump. And one sits there and wonders, how do you have that happen between male and female at the same time? The problem, again, is this idea of natural selection tends to preserve rather than to create. And therefore, why would there be anything that's going to be interested in this, especially if there is no background of that particular species existing prior? I mean, it just seems, what a fantasy world this seems to be coming out of. Solutions often arise from a subtle reformation of the question, not from a brute force collection of new information in an old framework. We avoid the excellent question, what good is 5% of an eye, by arguing that the possessor of such an insipid structure did not use it for sight. So Stephen Jay Gould, who also came up with this idea, also identifies the fact is that if you're looking for natural selection, why would this be selected? What then would be used? What would it be used for in order to preserve it for long enough to be finally used? If it was useful for something else, why would it continue building toward an eye when evolution has no goals, no purpose-driven direction? In other words, if it's functioning, why break it? And that would tend to be more in terms of natural selection. That would tend to be more in terms of random. Why do you preserve something or change it into something further on down the line? Again, there is that innate sense of a plan or direction that evolutionists seem to carry in the background. Particularly sexual reproduction tends to eliminate harmful mutations. If so, since evolution is goal-blind, why then would helpful mutations not also be likewise eliminated? I mean, again and again, you have the sense that evolution is saying, but it has the ability, the capacity to reason, the capacity to plan, the capacity to have a direction. And yet, if it's random, if it's truly random, that can't happen. Moreover, LGT, lateral gene transfer, in other words, you have two cells side by side and they go and they switch DNA information. Between such diverse organisms would itself require an incredibly complex and coordinated information system to facilitate such a transition. In other words, it isn't merely just take material over. Somehow it has to get integrated. Somehow it's got to be part of that error checking routine. Somehow it's got to be part of the protein building routines. Somehow it's all got to then be incorporated into this very precise machinery that already exists without it being there. Now somehow bringing it in, and somehow it has to be integrated. It's easy to come up with a fantasy. It's hard to make that fantasy work. Experimental science is hard-pressed to find examples of a random mutation that produces new information where neo-Darwinism requires many. Moreover, we see an inexorable trend of genetic deterioration caused by near-neutral mutations that will eventually lead to the extinction of all multicellular life. Even with systems that are designed to produce a certain amount of variation, which may involve a measure of LGT, there are fixed limits to design variation potential, because for life to work, it must be able to maintain fundamental genetic and cellular stability. So you just can't keep cluttering, throwing stuff in and expecting that somehow it's all going to work and everybody's going to be happy. Therefore, not even this can stave off forever extinction by genetic deterioration. Molecules demand evolution expects the exact opposite of what we see happening in biology. So it's a seemingly impossible concept. As the saying goes, he kept fixing it until he broke it. In other words, if the cell is constantly getting other stuff in there, fixing something that really isn't broken, finally you get to the point where it does break. When does the sheer weight of even neutral mutations finally break the cell? The line in the sand. How evolutionists treat the data. Richard Lowenton wrote, "We have a prior commitment, a commitment to materialism. Moreover, that materialism is absolute, for we cannot allow a divine foot in the door." It is not science. It is the philosophy that says it can only be this way. Scott Todd said, even if all the data point to an intelligent designer, such a hypothesis is excluded from science because it is not naturalistic. This kind of an attitude we have seen many, many times. We've seen in terms of redefining fossils. so that you can't have something that would shake up the wave. We have seen in all kinds of different places throughout this whole course of the fact that the reason why these things that are excluded is not because of the science, but because of the philosophical basis, philosophical commitment of certain scientists. On the other hand, when one evolutionist faced the data, what I think the DNA material has done is that it has shown by the almost unbelievable complexity of the arrangements which are needed to produce life, that that intelligence must have been involved in getting these extraordinary diverse elements to work together. It's all a matter of the enormous complexity by which the results were achieved, which looked to me like the work of intelligence. Although I was once sharply critical of the argument to design, I have come to see that when correctly formulated, this argument constitutes a persuasive case for the existence of God. This is the late Anthony Flew, an Oxford philosopher, once the world's leading atheist, but DNA helped change his mind. It's interesting how creationists accept all scientific data, while there are multiple examples of where evolutionists discard any scientific data that does not fit the model, no matter how compelling the data can be. Truly, as we said in the very first module, theory determines data. Or as it was said last time, theory determines truth. Hello, this is Dorbert's all-natural holistic tech support. Try stuffing tree bark up the CD drive and meditating. No, of course it won't damage anything. It's all natural. For the lemmings, are you sure this is the way to the beach? It's a shortcut. You see, Ricky, we learn from our mistakes. No wonder you're so smart, Dad. Yeah. All right. Assignment. You've got 2 readings, chapter 8, 10, which is 93 pages. So this is going to be another one of those 2-week type of deals. For this week, 2 online articles. Then for next week, 2 more. And then also the major project response. Thank you. I hope this is something in which you have really come to appreciate what has been built into the DNA, and maybe I have given you a chance to see the absolute awe I have in this molecule. Thank you.